Okay, so luckily we got this to work, and so in general, when we talk about the membrane, obviously it's not over there alone. It has many proteins and that span the membrane, that cross the membrane from side to side, which we will refer to as integral proteins. This is also an integral protein. We have membranes that associate to the membrane or to membrane-bound proteins, but are not covalently linked to the actual membrane, or linked to, or do not interact directly with the membrane, which we will call peripheral proteins. And there's also a subtype of proteins that are lipid anchored, meaning that they have a tail that forms this interaction, both hydrophobic and van der Waals interaction, between one of the sides of the membrane, and we'll call them lipids that are anchored to the membrane with this small anchor. So mainly, most proteins that span the membrane have a very defined structure, and most of the time the components or the motifs that you will find that are spanning the membrane are alpha helices. Or specifically, when we talk about channels, a lot of times it will be 7 membranes spanning alpha helix structure, as in this protein. And can you guess why specifically alpha helix is good for this? Yeah.
Because it has like the backbone is the outside and it's lipophilic.
Okay.
So yeah. So and that fits into the fat surrounding of the membrane.
So what is when we talked about I think you got it right. We'll just term it in like more exact terminology. When we talked about the alpha helix in general, one of the things that is unique about this helix is that in the backbone, okay, and these are all the side chains like I told you before, but the backbone of the peptide, all the hydrogen bonds, all the potential hydrogen bonds are actually occupied because of this structure. They're all they're all already in interaction, and thus the hydrogen bonds here, or the potential hydrogen bonds that can form between, let's say, this oxygen or this amine group with the hydrogen here, they're not free to interact with water, and thus, if you only choose, and thus the hydrophobicity of this structure is solely determined by the nature of the side chains of these amino acids. So if you take only hydrophobic amino acids, so this whole structure will be hydrophobic. As opposed to, I didn't bring an example here, but if we mentioned like beta sheets, in beta sheets you always have like these free or oxygen or amine hydrogens that are free to form hydrophilic interactions or hydrogen bonds with water. So in general, inside the membrane, which is highly hydrophobic, then you will have structures of proteins that are hydrophobic as well. And as I showed you before, this is just a more specific example. You can have also these anchors that are formed from hydrocarbons, which hydrocarbons, we mean just a long chains of carbons with saturated with hydrogens. And these anchors are normally formed in specific connections to specific amino acids of the protein. So you can have these kinds of connections, and you can also have different kinds of connections, which are through which is through a special kind of anchor, which is a sugar bound to hydrocarbon tails over here, which is called the GPI anchor. So we talked about a little bit about the membrane in general, and now we want to, and as I told you, the organelles that we have inside the cells are also defined by the fact that they're surrounded by membranes. And I will just go over the most basic structures that we'll find in any eukaryote cells. So the structure number one, as we talked before, is a plasma membrane, or this long cover or the sheet of the bilipid, phospholipid bilayer that covers the external part of the cell. You have then in all cells you have multiple mitochondria or mitochondrion, which is also surrounded by a double membrane, and this is as I told you, the power plant of the cell. You have the lysosomes, which are in charge of degrading worn out cellular membranes or proteins or extracellular proteins. The nuclear envelope, which here you can see really nicely, which covers the area where you have the DNA of the cell, and it's composed from two membranes. One membrane is the internal membrane, it defines the shape of the nucleus itself, and the external membrane, actually here you don't see it very nicely, but it fuses and forms the structure, which is the structure which will be the endoplasmic reticulum, which is this sac-like structure that is surrounding the nucleus, and actually this structure is all over the cell, not only in the nucleus, not only around the nucleus. The nucleolus, which we discussed in previous class, is a location. So this is like you would imagine like a compartment in a bacteria. Okay, it's a structure that is it's not bounded by a membrane, but it's a specific location inside the nucleus where you have the synthesis of the RNA. Okay, so in a bacterial cell you will have for all these different organelles you will have locations, but are not membrane-bound areas. For example, the mitochondrion can be like almost the size of a bacterial cell. So just to get into proportion of the difference in sizes between bacterial cells and eukaryote cells. So the nucleus is filled with chromatin, which is what we call is the mixture. Is like chromatin is a term to say that we have DNA and proteins that are packing the DNA together, and this is the site where all the genes are transcribed and the mRNA and tRNA are made. Like I said before, the endoplasmic reticulum, which like buds off and is part of the external membrane of the nucleus. It's in charge of this is here you do they did a separation between two types of endoplasmic reticulum. You have a smooth endoplasmic reticulum, which doesn't have these spots on it. Actually, it looks I don't know why they showed it. This is 7, so it's supposed to be like here, but it's wrong. I think they point toward this structure, but you see that in this structure you don't have these spots that we'll talk about later. And this is mainly in charge of synthesis of lipids and and detoxifying all sorts of compounds. Then you have the rough endoplasmic ER, which is generally like the ER, but it has these spots on them, and the spots are actually ribosomes. So certain proteins that need to be bound to membranes or need to be exported outside the cell, they have a special mechanism in order to do that. Is once I don't remember if you saw in the illustration that I showed you before about the ribosome, that the peptide is starting to come out, and then there's another protein that binds to the peptide that starts coming out and attaches it to like a membrane structure. Maybe I'll show you. Actually, I can't show you because I closed it. So never mind. So proteins that needs to be shipped or need to be inbound in a membrane, they tend to cling. The ribosome, once the peptide starts coming out, there are special proteins that know how to recognize a signal from the peptide itself, and then they carry this whole complex of the ribosome and mRNA, and they attach it to this membrane, and then the protein, when it's formed actually towards inside this area. Okay, and then there's a vesicle that is formed, and this vesicle is later shipped to a structure, to a very unique organelle, which is called the Golgi, complex. And this complex, it receives and sorts proteins that needs to be secreted outside the cells or embedded into the membrane, as well as also certain lipids are synthesized inside the Golgi complex. And here you can see these vesicles that are like budding out of this structure here and getting either secreted or fused to the membrane of the cell itself. And many times they contain proteins that needs to be expressed or need to be located in the extracellular domain of the cell. So this is not a very good picture, but in general, we can say that there are like two unique structures that are unique to neurons, and we can say, like, in a sense, they're like organelles of the neuron, which is not very exact. But in general, you have the synapse. As you know, each neuron has about 10,000 synapses on average, and this structure is like this. The structure that I showed you in the beginning of the class, it has this like button or spine membrane formation with these small vesicles that are ready to release neurotransmitters. And also the axons of the cells are very specified compartment. You can call it like a compartment of the cell that is built to carry electrical signals over long distances. So apart from the as I told you, now we're going to focus on more vesicle type of organelles, which we call they're like the lysosomes. So in each cell you have multiple vesicle organelles that are specified that have specific functions. So for example, phagosomes, which are especially common in the immune system, they take up all sorts of material from the outside, or microorganisms from the cell exterior. They take it inside and then they digest it. Endosomes take up soluble molecules that the cell collects from the exterior part. Let's say it needs to get nutrients and all the materials that the cell needs to survive. So it picks it up. It picks this nutrients from the cell exterior. Lysosomes, like we talked before, are acidic organelles that contain a lot of degrading enzymes. And there's also a process which is called autophagy, in which old organelles are being recycled inside the cell. Like an organelle that is already old and does not function in a good way can be readily digested. So in the lysosomes, you can find multiple types of enzymes that degrade each one according to the molecule. And it's very, the ases means that it degrades the letter that come before. So nucleases degrade RNA and DNA. Proteases degrade proteins. Phosphatases remove phosphate groups, mostly from peptides. And this is an illustration that summarizes this whole degradational processes. So in general, also this is the phagosomes, which are, like I said before, mostly common in the immune system, where immune cells catch foreign intruders or bacteria and encapsulate them and then degrade them. You can have also endocytosis, which is a process in which the cell takes nutrients and forms into endosomes, which these endosomes later come together into these lysosomes. Or you can have this autophagy vesicles, in which, in this example, you have an old mitochondria and it needs to be degraded. Then it's encapsulated in the membrane of the ER, and then then also this compartment is converted to be a lysosome. Okay, so in general, if you have all these foreign objects, it can be either nutrient that the cell needs, or like a hostile agent, or an old organelle. All these components can be recycled, and in the end they form lysosomes that degrade the proteins and recycle them for the use of the cell. So these are a few EM microscope images just to put everything in context of reality. So what is the structure? I don't know if you can see here, but could you tell me what is the structure here? What is this? This is a mitochondrion. Okay? You see this like a striped formation, which is caused because of it has these two membranes that are one membrane is outside and the second membrane is like formed of these curvatures. So this is the mitochondrion. What is this? This is the nucleus, correct? What is this? Here? Right? It's a nuclear membrane. These I don't expect you to know, but these are actually like vesicles that are to be secreted from the cell. And well, it depends on the composition of also how they made the staining in general. A lot of time you can have them black and you can have them. They made but they have, for example, they can have an environment that is different, and also different concentration of proteins and different. They also, one of the things that may happen here is because of the acidity, maybe the preparation of the sample, they might have like being degraded, and this is not actually how they look like. I don't think that they're more like that they should look like that. I think it's just a mechanism of the staining that they look like that. And in general, it's very hard to see here, but these are like very small vesicles that are related to the Golgi complex. And then you have the membranes of the endoplasmic reticulum, and in the end the plasma membrane and the intercellular space. There's another name saying the extracellular lumen or matrix that you have between cells. This is a close-up image. Again, it's actually not a very nice image, so I'll just jump over it. These are other images of mitochondrion, which is less nice than what we saw before. Again, this is vesicles of the smooth ER, and this is the rough ER. As you can see here, the ribosomes are like attached to the outer membrane of the rough ER, and the proteins that are being translated to the internal part of this structure. And as you can see from here, the cell is generally like a total mess, right? Like everything, it's not like organized and nice like in the caricatures, but it's like a mixture of a lot of membranes and a lot of vesicles and a lot of things everywhere. This is an example of a nucleus, and what is this signifies. This is just the nuclear lumen or the nuclear space. This is the area that we would call the nucleolus, where the RNA is. And the black spots here and the black spots here are something that we didn't talk about. But as I told you, DNA is in the cell in general, when it's not, although we have here the chromosomes that we talked about, they're not normally in the cell, and if the cell is not dividing, they will not be found in the shape that you normally are used to seeing chromosomes, in this like typical shape of chromosomes. They are spread out along the cells. But not all DNA is available for translation, and this is also one of the variability, or one of the reasons for differences between different tissues. In each tissue, you have different areas of the DNA that are available for for transcription and translation or expression of genes. And the areas that are unavailable are being condensed and packed in very tight formations. And this is what you see here as heterochromatin. Okay, chromatin is again a word to say DNA and proteins together. And heterochromatin is dense DNA and proteins. And what you see here, over here, all this area is what we call chromatin, which is loose DNA and proteins. So the DNA which is loose is being expressed, and the DNA that's being compact is being, you can say, suppressed. So in each cell, in each different tissue, you have different areas of the DNA or different locuses of the DNA that can be open or closed. And this will determine the genetic profile or genetic expression of genes that will be different in each tissue. Okay. No, in general, the fact that it's packed, it doesn't mean that the cell can actually change. You can like reprogram the cell, and this is what you talk about in stem cells. They have the ability, they have a higher flexibility or the ability to modify the expression of the genes. So this state, although we saw the heterochromatin here, it's not like in general, if we're talking about a neuron in general, then these will remain constant. But if you want to, for example, you have the stem cells and you induce a process that makes this stem cell like differentiate to a different type of cell, then some areas will open and some areas will close of the DNA. So it's a dynamic process. But in general, all the genetic material in all the cells is equal and is also present in all the cells. Okay. No? Stem cell can well, it's actually a very, very complex and a lot of it is unknown. Like this is what you're asking is the answer to it is like I'll have to give a course about stem cells in general to answer it, because it's a good question, but it's very complex. Why does a certain cell have the ability to transform itself to a different cell, and why do other cells don't? Like before Yamanaka that invented these that discovered that even you can take a skin cell and convert it into a neuron, people thought that it's not possible. Okay, that said, okay, once a cell is differentiated to a specific type of cell, then it doesn't have the ability to transform back to be a different, to reverse, to be like this omnipotent or pluripotent cell that can actually form, that can turn into other types of cells. So much of it is unknown, but it's also a part of large research in general. But here we're just describing like the fact that you have in any given cell, and that's true also in stem cells. Although it's true that in stem cells you will have a larger variety of cells of the chromatin that will be a little bit more that will be more open. But in general, what I'm trying to just describe here is the phenomenon that not all genes or not all regions of the DNA are available for active gene expression in a given cell or tissue. Okay, but if you want, we can discuss it. I can refer to like reading material if you want about it. But it's it's a very large topic. So this is a nice image of the mitochondrion, and here you can see very easily these membrane. These are called cristae, like the internalization of the inner membrane of the mitochondrion. And again, this structure, we're not going to get into that, but this structure is what helps the function of this organelle in order for it to produce the energy that the cell needs. Okay. So we talked about the fact that the membrane is like very fluid and very dynamic and everything moves around it, but the cells actually have to be very rigid, right? They have to have a specific structure, and they have to hold this structure. And the molecules, or you can say that the organ in the cell, or the organelle in the cell that is responsible for this structure is what we will call the cytoskeleton. So the cytoskeleton in general, we can divide it. It's made from these long filaments, or long filament molecules, that we can divide into three categories. The first category, which is the smallest of the filaments, is the actin filament. It's also called microfilament because it's the smallest. It's about 8 to 9 nanometers in diameter, and it has like a twisted formation. Also, all the microfilaments have these twisted, like rope-like structure, and it is composed mainly of a subunit that's called actin. So there's a small protein called actin, and the filament is actually composed out of multiple subunits of the protein actin. The next one, we're not going in the order of size, but the microtubule, which is the structure that you see here, is a hollow-like or tube-like structure that is 24 nanometers in diameter, and is made from two subunits that actually form dimers. It's alpha and beta tumulin, and these two dimer, and then they it's actually not a completely self-assembling structure, but they form this tube-like structure that also we saw an example of the motor proteins walking on this structure, and it also has a polarity, meaning that one side of it has a negative charge, and the other side has like a positive charge. The intermediate filaments are much less defined, meaning that you can have many proteins that are inside this family of proteins that we will call intermediate filaments, and they're intermediate because they're somewhat in the middle of the size between the microtubules and the actin filaments or microfilaments. And again, they're made from different subunits, and they're diverse in the nature of the proteins that assemble them. So these microfilaments, together with the membrane and the membrane-binding protein in general, they form the skeleton of the cell, like the structure and skeleton of the cell, which, as you can imagine from the name of it, they support the structure and they help the cell create the structure or the shape of the cell that it needs to in order to act. So this is an example of a fluorescent image from two cells, in this case, in which the microtubules are stained in green here, and the centrosomes, which is the location normally where the microtubules propagate or start from, normally located in one of the centers of the cell. And you can see, if you would see like a light image of this cell, you would see that this actually depicts the whole shape of the cell is supported or structured by these long filaments or long fibers. There is an example of a neuron which is grown. This is a cultured neuron, like a cultured hippocampal neuron. And here in this image, you can see, in this case, we have a microtubule-associated protein, or MAP2, which is specific to neurons, and it's located mainly. This is the protein that makes sure that the dendrites, that forms the dendrites and holds them in place. And you can see that it's highly enriched. This is the green protein. It's highly enriched in the dendrites. It's not enriched at all in axons. You don't see the axon of the cell at all. In blue, you can see nice nuclear staining. So this is actually staining of the DNA of the cell. And in red, you can see, like in this case, there are very small actin filaments, which are actually the synapses, which are enriched in the synapses of the neuron. Okay? You're right, but this is I'm talking about MAP2 in general. Like there are different types of microtubules. Okay, then in MAP there are microtubules that are also inside axons, but this specific microtubules, this microtubule protein specifically is enriched in dendrites. Okay, so many times, also one of the things that I'm doing in my research is trying to purify different compartments or different organelles of the cell in order to study the properties that are specific to these organelles. So how do we do this? How do we purify only parts of the cell that we want to study? Then first of all, one of the things that you can do if you want to like separate, there's actually before you separate the organelles of the cell, one of the things that you want to do is separate between the different cells. So let's say I'm only interested in a specific subclass of neurons, and I just want to take these neurons and then study more in depth what is going on specifically in these types of neurons. So in order to do that, we have this machine that's called a FACS machine, or fluorescence-associated activated cell sorter. And what this machine actually does is that you take a mixture of cells, okay? You suspend them in a sort of medium, so they're all floating around in the liquid, and then you pass them through the nozzle, and this nozzle actually creates like droplets, and each one of these droplets contains one cell, and these cells you separate between them because you previously did some kind of manipulation on these cells that makes only the specific cell that you want to look at fluorescent, or they will express a fluorescent protein in a specific color. And because you know the specific color, then you can, when the specific cell that you're interested in the droplet is passing through a laser, then you can apply an electrical force or charge the specific droplet that's carrying the cell that you're interested in, and then because of these deflection plates over here, it can deflect the cells to either location that you want to collect the specific cells that you're interested in. Again, first you have the cells. Let's say I'm interested in my case, it's like the cholinergic neurons in the brain, and I've created like a mouse line that expresses just a red fluorescent protein in these specific cells, and now I'm taking the brain of the mouse in my case and separated the cells into like an aqueous solution, and now all the cells are like floating in the solution, and I want to just take the red fluorescent cells. So I pass the solution through this special machine, and this machine, because it formed these very special droplets that each droplet contain only one cell, then it has a procedure where it analyzes the droplets. It sees whether this droplet is fluorescent or it carries a specific fluorescent protein that I'm interested in. Then, according to that, it can give it a specific electrical charge. Okay, so let's say it charges all the molecules that are red. It charges them in a negative charge, and then this droplet passes through an electrical field, and this electrical field separates physically the droplets that I'm interested from the other droplets. And thus I get only the specific cells that I'm interested in. Okay? It's a little bit... Okay, so just an example of how this looks like. For example, these are, like I told you, these red cells that are now grown. These are cholinergic neurons grown on a plate. And you have the red cells, which are cholinergic, and the other cells, the other neurons, which are non-cholinergic. And the cholinergic cells express a red fluorescent protein. And later, when I pass them through this machine, I can segregate them to different populations, like in this example here. Like according to the fluorescence that they show in the red, which is this axis, then I can separate them to different groups. And when I give the order to the machine to just take the cells that go into this category of fluorescence, meaning that if you detect cells that are fluorescent like this, then keep them, otherwise throw them away. Okay? So after I have the cells that I'm interested in, then I want to separate the specific organelles of the cells. So how do I do it? Because one of the most simple technique is by simple centrifugation. So if we have, I'm not going to teach you here about like Stokes' law that actually describes the tendency of a particle to precipitate in a liquid. But in general, if we take the cells, then like rupture them, and then move it to like a filter to homogenate. And when I say homogenate, I mean that this now contains all the different organelles of the cell. Then I can precipitate them in different speeds and get different structures. And also logically, if the structure that will precipitate first will be the heavy structures or the large structures. So one of the largest structures in the cell is the nuclei, or the nucleus of the cell. And if I take this homogenate and centrifuge it in 600 G, which is not very, it sounds a lot, but it's not very strong, then I would get here a precipitate of the nuclei. If I then take the supernated, or the upper part that is precipitate, and centrifuge it faster in 15,000 G, then here I will get precipitate of mitochondria, chloroplasts, we didn't talk about, and large, like lysosomes. Next, if I want to precipitate like smaller organelles, then I would have to centrifuge it faster and faster. In the end, the maximum the smallest units that I can precipitate with this procedure, I have to go to 300,000 G, which is pretty fast. And then I will get a precipitate from even small subunits of ribosomes. Okay, and this is like the minimal structure, the minimal unit that you can isolate through this procedure of centrifugations. Okay, so again, the centrifugation is if I centrifuge faster, I can precipitate organelles or particles which are lighter and smaller. Okay, centrifuge in a slow speed, then you'll get only the large structures. Centrifuge in a high speed, then you'll get like small structures. If you already took out the larger structures from before. So first, as I told you, you rupture the cells, then you get all the organelles inside, and then you do differential centrifugation in order to fractionize and get different compartments of the cell. Another method to do that is doing it according to density. For example, different particles have different density. Let's say if you have a specific, and with this we'll finish. Let's say I have two particles that I want to isolate, and one of them is filled with material, and one of them doesn't have so much material. And let's say they're the same size, although I'm not very good at drawing. Okay? So they're in the same size, but they have different densities. Okay? So one of the things that I can do is not simply centrifuge them in an aqueous phase, like in this example. I can form this gradient of densities, or different densities, and according to a specific density, if I apply enough force, then the organelles will migrate through this gradient, and only settle in the specific area where their density that is equal to their density. Okay, so this is done mostly with just using sucrose or mixing sucrose with water into to form this dense solution. And according to the specific density that we know, let's say I want to isolate mitochondria, then I form this density that has one layer that is more dense and one layer that is less dense than the mitochondria, and then all the mitochondria we're going to settle between the two phases of the density. Okay, because you can compare it like to oil and water. Oil is less dense than water, and then it will float on the water. So the mitochondria is more dense than here, but it's less dense than this phase, and it will just float on this phase, and thus I can get certain organelles according to their density. So just last slide is that this technique is also been used to isolate. Let's say I want to isolate synapses, or what we call synaptosomes. Synaptosomes are actually compartments that are made from the presynaptic and postsynaptic terminals that are like pinched off together. So if I just want to isolate that compartment and like study synaptic proteins or synaptic mRNA or synaptic properties, then again I form this gradient and I isolate and I put all the cellular homogeneate over here, but just in a specific, but because I chose a specific density that is fit to synaptosomes or to synapses, then I can isolate only synapses from this band after centrifugation. This is not important. Okay, so I'll repeat it. What you actually do here is that you take the organelle fraction, or what you want to isolate, then you apply, normally it's a huge force, like 200,000 G or something like that, because you want you want all the particles to migrate through the density gradient, and the particles will stop according to their density. So denser particles will go lower, and less dense particles will go higher. And this exactly is the same technique as I'm showing you here, that you form these layers of density. This time it's not with sucrose, but it's a different material, but it doesn't really matter. And according to the density, you can isolate an area where you just have synaptosomes. And you can also isolate synaptosomes that are pure synaptosome, or synaptosome with membrane, synaptosome with myelin that are covered around them, and so on and so forth. Okay, so that's it for this chapter. Next chapter, we're going to start talking about ion transport through membranes and start getting into more neuronal functions that are unique to neuronal functions of the cell. Thank you. And another thing, if you have questions, please ask, because you're not asking enough questions. Like I think there's just one asked me a question in the email. And normally it's a bad sign. So if you have any questions, feel free to ask. Okay? כן, כן. So.